Hello, and welcome back to day 5 of genetics. We did it, guys. We made it through a full week. It has definitely felt like a full year, but nevertheless, we made it to day 5. So, same as always, I'm going to go through today's worksheet with you so that you are able to fill yours out. Now, the thing about today's worksheet is that it requires you to use a computer simulation. But that simulation does not work, unfortunately, on your phones or on either Google Chrome or Safari. It only works, for whatever reason, on Internet Explorer and Firefox, because apparently it is still stuck in the 1800s. So that's the purpose of this video. I'm going to go through this simulation with you so that you are able to use that information to fill out your worksheet. Okay, so the very first process that we're going to cover is transcription. Now this is what we covered in, I believe, day 3's video, but this is going to be an overview of what we learned. So you have this text on your paper, but I'm going to read through it on this browser. So while you are following along with me, make sure that you are either underlining or highlighting important information on your worksheet. DNA carries the genetic instructions or code to produce proteins. The code is actually the sequence of bases in DNA. For example, ATA, GCGA is a code for part of a protein. The entire protein would actually require a much longer sequence of DNA bases. Whenever a protein needs to be made, the correct DNA sequence for that protein is copied to a molecule called messenger RNA, or mRNA. Since the DNA is so important, it is efficient to simply copy its code when needed and leave the original safe in the nucleus. And lots of copies can be made at any time. The process of copying a sequence of bases in DNA into a complementary sequence in mRNA is called transcription. It occurs in the nucleus and it is the first step in protein synthesis. So the instructions for this simulation right here tell us that a DNA sequence is shown below. The sequence is a small part of a gene that codes for a protein. We are to transcribe the DNA to produce the mRNA copy by placing the correct bases into position to make the mRNA transcript. Place additional symbols to fully label the process. Alright, so because this is the first step in protein synthesis, we know that this is found in the nucleus, so this very first strand is going to be our DNA. Another way to know that it is DNA is because there are T's present in the code. Whenever there is a T, that is DNA. And because we know that this process is transcription, I'm going to place this label right here. We know that transcription is when we take DNA and we convert it into an mRNA strand. So that means that our second strand right here is going to be mRNA. Now, we all know our base pairing rules by now. We know that A binds with T and C binds with G. However, when we have an RNA sequence, T is replaced by U, or uracil. So we're going to go through this strand together. We know that if we have a G in DNA, we're going to have a C in mRNA. Okay, next up we have an A in our DNA strand. So that means we're going to have a U, not a T, but a U in our mRNA strand. And same thing, we have another A in DNA, which means we have a U in mRNA. Now our second codon in DNA is CTC, which means that our codon in mRNA is going to be GAG. And finally, our last codon right here is AGT, which means that our opposite codon is going to be UCA. Now I'm going to use this button right here to check my answers. Alright, and let's check it out. So, first strand was DNA, second strand was mRNA, and this process is known as transcription. We know that if our first codon is GAA, our mRNA sequence is going to be CUU, GAG, UCA. Now you have two options for getting this information onto your notes. The first option, which I would recommend, is simply screenshotting this picture on the video. So you have got, if you're on a MacBook computer, I know that you can do Shift-Command-4 and then just circle that image and screenshot it like that. That way, when you go over to the Finder or on your desktop, you will see that image right there, and then you can simply scroll it over and insert it directly into your document. So that is one way to get that information into your notes. However, another way is you can click on the image that we originally gave you and copy it. So click Command C. Then you're going to click on this Insert button at the top and go down to Insert Drawing and click New. As soon as you get to this screen right here, you're going to click Command V or paste that image directly into this drawing. And then you are able to insert text boxes over the image and write down and fill in each of those boxes. So that way, when you click Save and Close, your text boxes appear directly on top of that drawing. So those are two images if you are on a computer. If you are using your phone, you should still be able to take a screenshot and insert it into Google Docs. It'll just look a little bit different, but I figure you guys will know how to do that. If not, just send me a message on Remind or on Google Classroom and I can help you get it sorted out. Okay, now the next part of this process is asking you a few different questions. Now, this part you are actually going to go through on your own. I want you to figure out the answer to these questions and bring your answers with you tomorrow to Zoom. But don't worry, because you have the ability to check your own answers on this website. So if you click the website on your phone, although you can't access the drag and drop options right here, you can access these questions right here, which means that if you type in your answer, you can check your answer and see if you are correct. That way, if you are wrong, you know to choose a different answer, and if you are right, it will tell you you're right. So let's go through number one. I'll show you guys what I mean. Number one says, which base in RNA is replaced by uracil? We know that U replaces T in our original DNA strand, and that T stands for thymine. So I'm going to check my answer, and it tells me that I'm correct. So then using that information, I'm going to go back over to my document, and I'm going to write down the word thymine on my answer sheet. So I want you guys to do that for all of these questions on your own, and once again, bring your answers with you to tomorrow's Zoom call. Okay, next up, we just finished transcription, now it's time for translation. Now that an mRNA copy has been made, the DNA zips back up and can be used again later to make more copies. Remember, the mRNA copy is also a genetic code for how to make a protein. The code was originally copied from the DNA, which is much too valuable to be used itself to make the protein, so a copy was made called mRNA. The mRNA moves into the cytoplasm, where the code will be translated on a ribosome by transfer RNA, or tRNA, molecules. Each tRNA carries one specific amino acid, which will be needed to build the protein. Since a protein consists of a precise sequence of amino acids, the right tRNA must insert its amino acid into the chain when needed. But how does this happen? On one end of a tRNA molecule is the amino acid. On the other end is a set of three bases called an anticodon, for example, CGA. The three anticodon bases use complementary base pairing with three mRNA bases called a codon, for example, GCU, and if they fit, this is the correct tRNA molecule and therefore the correct amino acid. See how the tRNA anticodon, CGA, is complementary to the mRNA codon, GCU? And note that both tRNA and mRNA have uracil instead of thymine. Since the tRNAs are essentially reading the mRNA code by binding to it and bringing in the proper amino acids to build the protein, this process is called translation. Alright, so for this next drag and drop sequence, the instructions tell us that the mRNA sequence that was transcribed in the first step of protein synthesis is shown with three tRNAs below it, each of which carry a generalized amino acid in this problem. Place the correct bases in a position to make up the tRNA anticodons. Place additional symbols to fully label the process. Whew, I need a sip of water. Alright, so we know that from our last process that ended with transcription, we took DNA and we turned it into mRNA. Now translation, this second part of the process, picks up right at the end of transcription, which means that our first strand that we're dealing with right here is mRNA. Now, since this process is translation, I'm going to fill this label in right here. And we know that in translation, the end goal is for mRNA to create proteins. However, that mRNA needs to be read or translated by tRNA before that can happen. So this second strand right here is going to be tRNA. And that tRNA, those tRNA codons or anticodons are going to match up with these mRNA codons based on those base pairing rules again. So similarly to how we did DNA and mRNA, we're going to use the same base pairing rules for mRNA and tRNA. So our first mRNA codon is CUU, which means that our tRNA anticodon is going to be the opposite, which is GAA. Similarly, right here, our first mRNA or our second mRNA codon is GAG, which means that our tRNA codon tRNA anticodon is going to be the opposite, which is CUC. And finally, our third codon is UCA, which means that our anticodon is going to be the opposite, AGU. Now, I told you guys in yesterday's recording that tRNA is shaped like a T, and on one end we have the amino acid sequence, on one end we have the anticodon sequence, and on the other end it's carrying the amino acid. So I like to think of the amino acids it's carrying as like a little backpack, because it's carrying those amino acids and it drops them off where they need to go. So I'm going to illustrate that with these labels right here, because on our tRNA molecules we have the anticodons on one end and the amino acids on the other. So let's check my answers and see if I'm right. And there it is. Alright, so first strand is mRNA, second strand is tRNA. They're each carrying amino acids. This is the process of translation. And once again, the anticodons of tRNA are going to be the opposite of the codons in mRNA. So our tRNA sequence was GAA, CUC, and AGU. Once again, I would recommend taking a screenshot of this and importing it into your notes. So once again, Shift Command 4, take a screenshot, find that image in your recents, and drag it into your notes. Another option again is to copy this image, do Insert Drawing New, paste that image into this drawing, and then use text box to write the answers. So then when you save it, your text is safe on the screen. Alright, similarly to what we did with transcription, I want you guys to answer these questions on your own. However, you can use this website that we've provided you to check your answers. So if you type your answer on this website, you can check it and see if you are correct. So make sure you fill these out tonight and bring them with you to tomorrow's class. Now, the moment you have all been waiting for, I'm sure, we're going to bring all of this together to show you what the entire process of protein synthesis looks like. So, protein synthesis. Now it's time to put everything together and look at the full process of protein synthesis, transcription, and translation. This begins with DNA in the nucleus and ends with the final protein in the cytoplasm. To do this, we need to be able to read the genetic code just as tRNA can. Since each 3-base sequence, or codon, of mRNA codes for one amino acid, we look up the amino acid in a codon table. We read what the mRNA codons are coding for. To use a codon table in general, start with the first mRNA base in the codon and find the first column of the table. Then find the second mRNA base and find the second column. Your choice of amino acid is now constrained to the area in the shared region of column 1 and 2. Now find the third mRNA base and follow it to the specific amino acid. Note the 3-letter abbreviation of the amino acid. So for example, HIS stands for histidine. Now here are your instructions for this next part. The DNA sequence below codes for a small portion of a protein. Transcribe the DNA to produce mRNA and translate the mRNA using tRNAs into the correct amino acids. Then identify the protein. Refer to the codon table and amino acid sequences. So this is the entire process of protein synthesis, starting with transcription and translation. So we know that the very first step of this process starts with DNA. Remember, we find DNA in the nucleus, and we are going to use that genetic information to eventually make proteins down here. So I'm confident that the very first strand is going to be a DNA. We also see that T's are present in this sequence, meaning that it is DNA. Now, the first step, known as transcription, is when we turn DNA into an mRNA sequence. So I believe that the second strand is going to be mRNA. So let's go through these codons. The first codon of DNA is CCC, meaning that the codon of mRNA is going to be the opposite. So it's going to be G, G, and G. The second codon of DNA is TAG, which means that the codon of mRNA is going to be A, U, C. And the third codon of DNA is CAT, which means that the codon of mRNA is going to be G, U, A. So now we have our mRNA sequence. The next step is going to be called translation. And that is when tRNA is going to read this sequence and then drop off the appropriate amino acids. So I'm going to label our next strand as tRNA. Now the way that tRNA is able to figure out which strand it needs to attach to is through those base pairing rules. So if our codon of mRNA is GGG, that means that our anticodon, remember when we're talking about tRNA we say anticodon, is going to be the opposite. So our anticodon will be C, C, C. And remember that tRNA is shaped like a T, and it has amino acids on one end and the nucleotide bases on the other. So I'm going to say that this label right here stands for amino acids because it is carrying that amino acid and is waiting to drop it off. All right, our second codon of mRNA is AUC. So our anticodon of tRNA is going to be U, A, G. Once again, it is carrying that amino acid, ready to drop it off and make a protein. Finally, our very last codon of mRNA is GUA, which means that our anticodon of tRNA will be the opposite of C, A, U. Once again, it is carrying an amino acid. All right, so when we combine all of those amino acids together, we make something called a peptide. And we will get into that definition later, but right now you just need to know that it is a sequence of amino acids. But now the next thing this simulation wants me to do is figure out exactly what amino acids this sequence codes for. So I'm going to pull up our handy dandy codon table, and we're going to figure out what amino acids are present. Now this is the table that a lot of you guys were scared by on day 4's recording, but don't panic because I'm going to show you exactly how to read this table. But one important thing that we need to note is when you are using this table right here, you do not use your tRNA sequence. You are going to use the mRNA sequence. So I wish I could draw on the board right now, but I can't, so I'm just using my cursor and hoping to emphasize that when you use this table right here, you're going to use the mRNA sequence, not tRNA, but use mRNA instead. So our very first codon that we're looking for is GGG. So I'm going to go over to my amino acid table, and the first thing you do is you find the first letter of that sequence on the left-hand side of this diagram. Now our first letter was G, so I'm not going to use this one, I'm not going to use this one, I'm not going to use this one. I'm going to use this section of our codon table because our first letter is G. Our second letter is also G, so then I'm going to look up here at the top. I'm not going to use this row because it has a U. I'm not going to use this row because it has a C. I'm not going to use this row because it has an A. I'm going to use this row right here because it has a G. So now I know that I'm going to be using this section of the table. If you hear any noise in the background, it's because my cat is attacking my window, so I'm sorry about that. So you're going to be using this last row of the chart. Okay, let me start that over. We've now narrowed it down to this section of the table right here, because our first letter is G, our second letter is G, so now we know we're going to be focused in this section right here. Now our third letter is also G, so you're going to come over to this side of the chart. We're not going to use this one because it says U, we're not going to use this one because it says C, we're not going to use this one, but we are going to use this row right here. First letter G, second letter G, third letter G. That means that we are going to be coding for the amino acid glycine, or GLY. So I'm going to find glycine down here, and I'm going to insert it right here. All right, our second codon is AUC. So I'm going to go back to our chart, find first letter A, second letter was U, and third letter is C. So I'm going to follow this over here, and our amino acid is going to be something called isoleucine, or ILE. So I'm going to drag ILE over here. And finally, our last codon we're going to be using is GUA. So once again, I'm going to go back to our amino acid chart. First letter is going to be G, second letter is going to be U, and third letter is going to be A. So I'm going to follow this row of the chart over here, and the last amino acid that's being coded for here is valine, or VAL. So I'm going to find valine right here and place it right there. Now, when we have these three amino acids together, we know that we're going to end up building a protein. That is our end goal. This entire process is talking about protein synthesis, which is talking about making or creating proteins. So now, we will probably never ask you to do this. However, we're going to get some extra practice. We're going to figure out what protein we're actually coding for. So I'm going to look at this chart right here, and this gives me two options. We're trying to figure out if the protein being made is hemoglobin or insulin. So our amino acid sequence is GLY, ILE, and VAL. Now, looking at this chart, do you think that we're coding for hemoglobin or insulin? I hope you guys said insulin. So now I'm going to drag insulin over here because that is the end goal, or the end protein being made. So I'll check my answers. Okay, there's not a place to check my answers, so I'm just going to trust that this is correct. Once again, I would say screenshot these final results, and then use that image and drag it into your notes. Okay, and finally, last but not least, you have got 4 questions that need to be answered. Once again, you can check your answers on this website, so click on the link in that document and use this website to check your answers. Finally, the last thing that this worksheet tells you to is to take the end quiz. Now that quiz can be found at the top right corner of this website. You can access this on your phone once again. The only thing that you can't do on your phone is these drag and drop things. So, go over to the quiz, and I want you guys to read through each of these questions and take the quiz on your own. Now, when you click when you click submit, let's see, I don't think I'm getting any of these right. When you click when you click submit, if you got an answer correct, there will be a blue check mark next to it. If it is not correct, you will not have a blue check mark. So I want you guys to keep taking this quiz until you get 10 blue check marks, meaning that you got every single question right. All right, and that is all I have for you today. Once again, make sure you are putting your answers on this Google Docs worksheet and submitting it in Google Classroom. Day 5's worksheet is due Friday night at midnight, but make sure you complete it before you come to Friday's Zoom class. If you have any questions, write down those questions and we will answer them over Zoom tomorrow. But other than that, have a wonderful day, and I'll see you guys tomorrow.